My name is Randi Vita. I work at the La Jolla Institute, and my talk is a structured model for immune exposures. Immune exposures are vaccinations, infectious, allergic, or autoimmune diseases, or transplantations that perturb the immune system. Knowledge about them is important for developing diagnostic tools and therapeutic interventions. There's a growing need to standardize how they are described, as increasing numbers of resources try and capture this type of data, and they want to be interoperable and machine-readable. I work at one of those resources, the Immune Epitope Database. This is a free online resource of experimentally derived epitope information that relates to allergic and infectious and autoimmune diseases and transplantations. We have more than 99% of all published data demonstrating the recognition of an immune epitope by the adaptive immune system. This amounts to more than 2.5 million experiments that were manually curated from more than 21,000 references. We've had the need to represent immune exposures from the start that covered all types of hosts, not just humans and laboratory animals, but any species who has a published immune response to an epitope, and all the disease types that are relevant to the IDB scope. The data in the IDB starts back from the beginning of PubMed, and we have data from as early as 1952, and we remain current with the literature to the modern day. We wanted to see if the way that we had been capturing immune exposures would work as a standard across other resources. So we became involved in the data standards for the HipC project. This is a human immunology project consortium. It's a collaboration between a number of centers that are aimed at performing large-scale human immunology studies, and they focus on profiling the human immune response to infection and vaccination. So this project also had a need to capture immune exposures. Much of the data from HipC ends up in IMPORT, the Immunology Database and Analysis Portal, another NIAID resource that specializes in capturing human immunology studies. They used structured data fields for their experimental data, but they used free text fields to describe the metadata on the cohorts, which includes the immune exposure information. So initially there was no standardization, just free text fields. And without standardization, it was difficult to query and compare across the different studies in IMPORT, and impossible to compare across other resources in what was in IMPORT. The types of information captured in these free text fields included this example here: subjects from an endemic area that tested positive for antibodies against dengue 2 based on serology. This is quite a bit of words, and a different researcher might describe this exact same cohort using different terminology, making it very difficult to break down what was in this study versus in another study looking at cohorts who also had dengue. So what we did was took the critical elements of the immune exposure as described in the IDB and laid them out for the IMPORT research project. So the exposure process was the type of process to which the host became exposed. It could have been a vaccination, it could have been an infection. The exposure material is what the host was exposed to. So it could be the dengue virus, or it could be a vaccine. And then if the exposure process resulted in a disease, what was the name of the disease and what was the stage at which the cohort was studied. Using those critical elements, we were able to create this list of immune exposure requirements and have these three main categories of administration, disease, and exposure without disease. These exposure processes cover all of the data within the IDB project and also all of the HipC data within IMPORT. The administration group, because something is administered, requires that exposure material be present. The disease group, because this is a group having diseases, requires that there be both a disease name and a disease stage. And the group of exposures without disease require what the group was exposed to, but you don't need a disease name or stage, as there was no disease name or stage. We took those critical elements and mapped them to ontologies. For the process types, we largely used the OB, the Ontology for Biomedical Investigations, and this allowed us the opportunity to incorporate logical validation to exposure materials and the disease state and stage. If the exposure material was a vaccine, we used the vaccine ontology. And by doing so, we have access to the different subunits present within a subunit vaccine. And for multivalent vaccines, the different organisms, say the different strains of flu that are present in each flu vaccine. And the exposure materials largely are derived from organisms, and so we have links to the organisms by using NCBI taxonomy, which gives us additionally links to the sequence information for those species. And if there was a disease, we used the disease ontology, and this provided us with logical links between the NCBI terminology and the disease that was caused. For example, malaria is caused by Plasmodium falciparum. If the exposure material happened to be the dengue virus, it would not be able to cause malaria. And if there was a disease name, we also require a disease stage, and we use the ontology for general medical sciences to standardize this terminology. And once we were able to represent all of those key elements using ontology terms, we were able to map all of the data within import from the HipC cohorts to our model. And these free text descriptions are actual projects that were involved in this. So for example, adults receiving varicella-zoster shot, this is broken down to the exposure process of vaccination and the exposure material of varicella-zoster vaccine. These subjects did not have a disease. Hospitalized patients with hemorrhagic dengue fever, as the cohort description, can be broken into an exposure process of an infectious disease that's caused by the dengue virus. And that disease name is dengue hemorrhagic fever, as it is in disease ontology, and the stage of these patients is acute as they were hospitalized. And that example from the start, the subjects from the endemic area with antibodies to dengue, these are exposure with immunoactivity. The immunoactivity is those antibodies. And we know that they were exposed to dengue 2 because they have antibodies to dengue 2. And now by using the NCBI taxonomy, we can see that these two cohorts are related as they're both exposed to dengue virus, one a specific strain and one higher level. By mapping all of their terminology to ontologies, we were able to create automated validation between the different elements of the immune exposure. We did this by developing an open source automated validation tool that loads the data from import and also the relevant ontologies, and then applies sets of validation rules. These are some examples. We're able to make sure that fields that should be filled are filled and fields that should not be filled are not. For example, if there's an exposure process, there shouldn't be a disease. But if there is a disease process, there should be a disease. It also checks that the right type of ontology is used. For example, flu zone is a completely correct exposure material as an influenza vaccine, but it's not a process type, so it shouldn't be used as an exposure process. This validation tool highlights the cells that are invalid and gives tooltips to explain what the validation errors are, and it renders the output to HTML or Microsoft Excel format, so it's easy to use, and Import's been able to use this to improve their data set. So the current status is that the values we are using here were assembled by our team over the past 14 years, so we believe it is robust and stable and that it truly reflects the immunology literature as it stands. The HipC studies are now better connected as they've adopted this model, and we've improved their query and comparison capability. And we've been able to engage the representatives of those different ontologies throughout this process to harmonize the definitions between the different communities, but also to do a lot of term requests, as many of the terms that we needed to describe the HipC studies were not already in, say, for example, the vaccine ontology or the disease ontology. So by doing this procedure, we not only made our data better, we also improved the content of those ontologies. So what we're doing now is trying to expand to other projects within HipC and outside of HipC. Immune Signatures is a HipC project that is not in import, and we've adapted our model for them. And then ViewPathVV and other databases that capture immune exposure data, we're looking to collaborate with and incorporate our standards into their projects. And if anyone who's listening to this has need to standardize their immune exposures, we will be happy to hear from you. We want more people to use it because our goal is to create a public standard using our definition and rules, and more examples will make it a stronger and more accepted model. This work was done in the HipC Data Standards Group. Myself and Bjorn Peters from the La Jolla Institute. James Overton at Notion, he created that validation tool that's so cool. And at Yale, Kay and Stephen Kleinstein, and at Import, Patrick Dunn, all contributed to this project. Thank you very much.